So we left off the other day talking about capillary exchange of gases and nutrients and how we have 4 different routes that substances are going to take if they're going to either enter or leave the blood. So we talked about direct diffusion, which is a flashback to freshman year of moving from a high concentration to a low concentration. So this is how those respiratory gases move, like oxygen and carbon dioxide. We could also go with diffusion through our intercellular clefts, and all of our cells have these intercellular clefts except the brain cells, because we don't want just any sort of materials passing through our delicate brain cells, or we could have some serious problems. And the third pathway is diffusion through those fenestrated capillaries, and if you remember, fenestra is a window. So this is a very absorbent way for materials to move, so especially through those intestinal capillaries, because they're going to do a lot of that nutrient absorption, as well as filtration with our kidneys, because our kidneys are going to, they need to be very permeable to know when to hang on to sodium and water or to let it go. So those materials are going to be passing through rather quickly. And let me click here. And materials that can't pass through, if you remember Gandalf here, if you like Lord of the Rings, like I do, red blood cells are not going to be allowed to leave the capillaries. We don't want them to go, or proteins. So proteins are very large, complex structures, and we don't want them to get out of the blood. They need to stay there. So let's go. Alright, so here we have a picture of the 4 different types of diffusion. Through our direct diffusion right through that membrane, it's lipid-soluble molecules like oxygen and carbon dioxide. Or we can go through an intercellular cleft, so molecules that are a little larger can move through here. And this is through your process, this is the brain. It does not have these intercellular clefts, but every other cell has them. Or we can go through diffusion with a fenestra pore, and this is going to be very absorbent pathways, so intestinal or filtration pathways like the kidney. Or we can have transport with vesicles, so that's your bulk transport of your endocytosis or exocytosis, which requires energy. Diffusion does not. Alright, so here's our fluid, how it's moving through our capillary beds. So depending on which way that fluid's moving, all relies on pressure, which is like your concentration gradient. So blood pressure is going to make fluid and solutes go out of the capillaries, whereas osmotic pressure is going to pull that fluid into the capillaries. And the reason this happens is because blood has a higher concentration of those solutes because they have so many of those plasma proteins than the interstitial fluid. So that's why blood pressure is going to push the fluid out, and osmotic pressure is going to pull the fluid back into the capillaries. Alright, so here we still are talking about the movement here, how our blood pressure is higher than the osmotic pressure at the arterial end. So this is where blood is moving away from the heart on that capillary bed. And then blood pressure is going to be lower at the osmotic end, because this is the venous end. Think veins. So we're going to have to take that blood back to the heart, and we know the veins, they have those valves in them, so very, very low pressure. So this, this makes sense of everything we've been talking about with the aorta having the highest amount of pressure, and we just go down in pressure from there. So that fluid is going to move out of the bed, that capillary bed at first, and then it's going to be reclaimed at the venous end. But not all of the fluid is reclaimed by the venous end of that capillary bed, and the job of the lymphatic system, which although it's chapter 12, we may be skipping it for just a moment to get to digestive and respiratory, but the lymphatic system is going to actually help pull that tissue and reclaim it. So when you're a tiny little embryo, you started out with a simple just tube heart, and I tried to put some pictures on here. I don't know if we'll be able to see them very well. So there's that heart tube. And by week 4, or sorry, during week 4 that heart tube is developing. And by week 7 you're going to get that 4-chambered heart that we've come to know and love. And that's at the end of week 7. So after week 7 you're pretty functional. And there's only going to be some very minor changes after you get through week 7. And I'm going to hit pause on this, so we'll be back.